Hi there. Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world.
Hi, everyone. Thanks so much for tuning in. If this is your first time being here, welcome. We share inspiring stories of people on the front lines of life sciences bringing life-saving products to market. And if that sounds like you or you want to share your story, we'd love to hear from you. There's a form in the show notes here to apply to be on the show. If you have an idea or know somebody, please submit their name. We'd love to get in touch. Today's guest is Dr. Ian Taylor, the Chief Scientific Officer at Arvinas. He came to Arvinas after nearly 10 years at Pfizer Oncology, most recently as an early development team leader. His role there at Pfizer was cross-functional in drug development treatment and conducted phase 1 and phase 2 clinical trials for multiple assets and several cancer indications. He's had a whole career in oncology as well with Bayer Healthcare Pharmaceuticals and got his BA from Bowdoin College and his PhD in molecular biology and genetics from Harvard University. And Ian today talks about the mission of Arvinas and pioneering science to develop new transformative treatments for patients with limited treatment options or for whom existing treatments have not been very effective. He and his team work in targeted protein degradation. And Ian goes more into this, but basically tagging cells in a unique way to naturally degrade. He also goes into the advantages of this type of treatment, as well as the future of being able to treat what is now 80% of untreatable diseases and the possibilities of making treatments a reality for those who suffer from them. Let's bring him in. Ian, thank you so much for joining the podcast today. We're excited to have you on the show.
Oh, it's my pleasure. Thanks for having me. I'm really excited too.
Yeah, we are excited to talk to you about what you're doing at Arvinas and the field, a really interesting field and the technology that you're working on over there to treat previously undruggable diseases. But maybe before we get into that, just for those who may not know about Arvinas, tell us about what you folks are up to there and your company mission.
Yeah, so at Arvinas, we're located in New Haven, Connecticut, and our mission is to serve unmet medical needs, particularly in the fields of oncology or cancer and neurodegeneration. And we're expanding into other areas as well. And we do that by what is known in the field as targeted protein degradation. For us specifically, we use a technology called PROTACs, or proteolysis targeting chimeras. These are small molecules that are designed, to eliminate disease-causing proteins from cells. A lot of drug therapies that people may be aware of are largely inhibitors. They inhibit the catalytic function of a particular disease-causing target, like an enzyme, which is fine, but there's a lot of limitations there. Mm-hmm. It's hard to fully inhibit a protein if it's really overexpressed in a cell, say like in a cancer cell. Whereas our technology uses the natural way that cells degrade proteins or turn over proteins, because cells are constantly doing that. They're making new proteins, they're getting rid of old proteins if they're misfolded or they're mutated, or they just outlived their natural lives. So we're tapping into that system with a small molecule and degrading the protein, just completely eliminating it from the cell. And we think that has a lot of advantages, longer term benefits of keeping that cell from being expressed, lower doses of the therapy, different functions that proteins might have other than their enzymatic function. That leads into the undruggable space. And we're again applying that to oncology, cancer, and neurodegeneration, such as diseases like Alzheimer's or Parkinson's.
Yeah. And one thing I think is really interesting about what you're doing there is I think you used the term hijacking. You're using the body's normal function of eliminating these abnormal proteins. And I think that is something that will capture a lot of people's attention because it's something that's natural to the body. So how did you get into that type of technology and how are you advancing that?
Yeah, so this is technology that was spun out of Yale University. So our scientific founder, Dr. Craig Crews, was the inventor of PROTACs 20 years ago now. Was when we first published a paper on PROTACs. So he had the idea of if you could somehow bring the way cells normally degrade proteins and make it attack a certain specific protein that you wanted to degrade right now in this cell, that could be really powerful. So the way it works is a small molecule, PROTAC, binds to what we call a heterobifunctional molecule. It actually has multiple functions, and that's why we call it chimera. It's basically 3 structural features in one small molecule. So one end of the molecule binds to the target that you want to degrade, so the disease-causing protein. The other end of the molecule binds to an enzyme in what's called the ubiquitin proteasome system. That's the natural system that the cell uses to degrade, or at least one of the natural systems. There's actually multiple. And so what you're doing is you're bringing this workhorse enzyme in the ubiquitin proteasome system called an E3 ubiquitin ligase. You're bringing it right to the target that you want to degrade. And the E3 ubiquitin ligase's job is to add a ubiquitin chain of molecules. This is a tag that the cell recognizes. Oh, once this protein has this ubiquitin tag, the cell says, oh, this protein's now ready to go off and be degraded. So once the PROTAC actually brings together the disease-causing protein in this E3 ubiquitin ligase, the rest of the process is exactly how the cell would normally do it. The E3 ligase ubiquitinates the protein, that tag of ubiquitins the cell recognizes, and off it goes to what's called the proteasome, which is this collection of enzymes that chops up the protein into its component peptides and ultimately into amino acids and completely eliminates it from the cell. So it's very intuitive. It's really cool, actually, the way it works. Mm-hmm. And it actually does work. We can completely eliminate proteins from cells by treating with small molecules.
Wow. I think I saw on your website, which is fantastic, Barry, there's a lot of great videos that kind of show exactly what you're talking about here in a very visual way for us visual learners. But you said that I think 80% of the proteome today is currently undruggable, and your technology is going to be able to address like a lot of that with being able to target specific cells and tag them this way. So just curious, how with this huge potential with neurodegenerative diseases, cancer, like you mentioned, how is your team focusing on where to advance and continue to research this technology and what eventually gets in your pipeline?
Yeah, that's a great question. So the undruggable space. So you're right, 80% of the proteome, which is the collection of proteins in a cell, are considered undruggable. And for those of us who have been working in drug discovery for a long time, and for me, it's been a long time, there's always been this great collection of proteins that are clearly linked to disease, but just have been intractable to traditional small molecules. Mm-hmm. And we've had to move away from those over the course of time. And there's just a long list. Some of them are called these holy grail targets that, boy, if we would really be able to treat disease. And this has been something that I've focused on for a long time. It's really what attracted me to come to Arvinas, was the ability to go after these undruggables. Mm-hmm. And the reason PROTACs can do it is because really all you need to do is with a PROTAC is bind to the target protein and then bring that E3 ligase to it. And it only has to bind weakly. It only has to bind transiently, just long enough to form that trimer complex of the PROTAC, the target, and the E3 ligase. Get that protein ubiquitinated, and then it goes to degradation. You don't have to have any other function than binding, mm-hmm. weak binding and transient binding. But traditionally, with traditional small molecules, you would have to attack what's known as an active site, right? An enzymatic site, the way that protein is working. But some proteins don't have active sites. Sometimes those proteins' active sites are really hard to get a small molecule in. There's things like transcription factors that just bind to DNA, but drive transcription, particularly in oncology. That's a big problem. Inappropriate gene expression leads to inappropriate tumor cell growth, which leads to tumors. There's a whole group of transcription factors that and they're like bowling balls almost, right? You can picture it as a bowling. They're very smooth. There's not that many nooks and crannies. Where can you bind a small molecule to inhibit its function? It's largely impossible. But with a PROTAC, all you have to do is bind a little bit in one of those nooks and crannies, get that E3 ligase there, ubiquitinate, and can degrade. And so those are the kind of targets that we are looking at. And also, we're really attracted to the undruggables, but also where does degradation have an advantage over traditional small molecule inhibition or even inhibition by antibodies, so other therapeutic modalities. Certainly, there are a lot of successful drugs that are working. We want to make sure that we're attacking things where degradation actually has an advantage. So it's not just a me too, but by actually removing the protein from the cell, it has an advantage over any other modality.
Got it.
Yeah, and that's how we select our targets. That's how we prioritize our targets. That's how we prosecute our targets with that fundamental aspect, what we call like the tenets of PROTACs. We have a collection of where does degradation have an advantage over other modalities.
That makes a lot of sense. Okay. Yeah, I appreciate you explaining that. What are some of, like you said, with some treatments, you can have unintentional growth. What are potentially some of the risks involved with this type of technology, like perhaps binding to the wrong type of protein or something like that? What are some of the risks involved?
Sure, that's what people always focus on. It's several risks. We get non-selective degradation, right? We're degrading other proteins that you want to degrade. And that's something we obviously track during the course of our lead optimization program when we're developing the drug. We can track that using what's known as proteomics. You look at thousands of proteins in the cell and make sure you're only degrading the one that you're going after. We've actually shown that for our lead programs that are in the clinic, ARV-110 that's going after the androgen receptor for prostate cancer, ARV-471 that's going after the estrogen receptor for breast cancer. We've actually looked and say, okay, are we degrading anything other than the androgen receptor or estrogen receptor? The answer is no, at least for the large part of the proteome that we can look at. And so we optimize for that as we go. The other thing that people say as well, you're hijacking the ubiquitin proteasome system, so are you affecting its ability to degrade other proteins that would normally degrade, and then they'll build up and cause a problem? The answer to that is no, as far as we can see, and we've looked many times. And that's because our molecules are really potent. So we're only using a small fraction of the E3 ubiquitin ligase, for example. We're only using a small fraction of the total capacity of the proteasome. And so we've not seen a buildup of other proteins because we're hijacking, because the molecules are potent, and we're just using a fraction of it. So those are the main issues that are concerned about. Of course, when you're doing drug discovery, drug development, you're always looking for toxicity. So we certainly track that in the clinic in our patients, and we try to minimize that. And the other thing with PROTACs is we can use lower doses because of the way the PROTAC works. You don't have to overwhelm the target with a lot of compound. Yeah. Because it's an event-driven pharmacology, the event being the ubiquitination. It's not an occupancy-driven pharmacology like traditional small molecule inhibitors that have to occupy that active site, and you need an excess of small molecule to make sure you're constantly occupying the site. So that allows for lower doses, but it also helps with the safety profile.
That is fascinating, Ian. I can't think of, I might be so bold as to say that everybody listening to this podcast, and certainly no one in my circle, has been unaffected by somebody with cancer or neurodegenerative diseases. And so the idea of having something that is safer and allows the person to recover faster than a lot of these overwhelming treatments available today just sounds really exciting. This is really innovative, the cusp of this field moving forward into the future. So that's just an exciting. And one quick side note, that you mentioned a couple of the treatments in your pipeline right now. We'll link to this in the show notes, but there are, I love how your website lists a lot of the programs going on and their current phase, if it's research and where they're listed here. So I think, and some of the partners that you're involved with, I just love the transparency on your website. We'll link to that in the show notes so people can see more information about some of the treatments that you just mentioned.
Yeah, we actually have 3 compounds in the clinic now. We have another androgen receptor called ARV-766, which you'll see on the website. We actually have over 20 programs that we're working on at various stages, both in oncology, immuno-oncology, immunology we're getting into, and also neurodegeneration, as you mentioned. So that just speaks to the power and breadth of PROTACs. We view it as what we call therapeutic area. It can really be applied almost anywhere. We actually have a joint venture you'll see on our website with Bayer Pharmaceutical, called Bayer Healthcare, which is called Earth Bio, which is bringing PROTACs into the agricultural space.
Interesting.
So beyond human disease. And to be honest, one of the surprises that I've had over the course of my career at Arvinas, when I started a little over 5 years ago, we were essentially the only company doing PROTACs and targeted protein degradation. At least PROTACs is applied to targeted protein degradation. Now, there are so many companies doing it. Large biotechs like us, academics. I honestly can't think of a field that's exploded as much within 5 years, at least in the drug discovery space, as targeted protein degradation and PROTACs has. Because everyone recognizes what you've already said, the potential power of it, the advantages in terms of adverse events or toxicity. Now it's easier to name companies that aren't doing PROTACs or targeted protein degradation than are, because everyone is doing it. Literally, everyone is doing it. And that's been great for us as pioneers in the space, all these people piling in, but it's really been eye-opening. It's wow. and people are bringing it into other companies are bringing it to other therapeutic areas. Again, just speaking to the breadth and the potential power of the technology.
Yeah. So Ian, I guess I'm curious, let's take a second to look in the rearview mirror a little bit. And how did you get here in your career? So we went over your bio a little bit. We'll link to it in the show notes as well. But you were at Pfizer Oncology for a number of years. You worked with Dr. Harold Varmus, the Nobel Prize winner. And so just from some of the experiences within your career, how has your experiences led to where you got to today, first of all, and then just some of the ways that you approach your work today from the experiences early on in your career for those who are just starting out?
Yeah. Actually, you touched on it. I'll come back to it. You touched on it earlier in one of your comments. Yes, I got my PhD at Harvard, and then I grew up outside of Boston, so it was a nice area for me, nice to stay local. I went to college in Maine. Then, I was lucky enough to get a postdoc with Harold Varmus, who was at University of California, San Francisco at the time. He had basically just won the Nobel Prize, sharing it with Mike Bishop for discovering oncogenes, so cancer-causing genes, and spent 3 years there. And then he became the director of NIH, National Institute. And so he moved there, and I was lucky enough to move with him and finish up my postdoc a year at NIH. It was really very interesting. Just NIH is huge, down in Bethesda, Maryland. And at the time I was finishing up my postdoc, and I started to apply for academic jobs to become a professor. And I actually had a couple offers, but right around that same time, and I was deciding, I got a call from a headhunter for a job at Bayer Pharmaceuticals up in West Haven, Connecticut. And I said, oh, that's interesting. I didn't really know much about the pharma industry, but I went on the interview and went on a second interview and decided to take the job. Because a lot of what I was doing in Harold's lab, looking for new genes, applied to what they were looking for. So I told Harold I was going to take this job at Bayer, and he said, gee, I thought you were taking this academic job. And I said to him, Harold, I'm not going to win the Nobel Prize. I'm just not, like you. But I think the next coolest thing would be to be involved in a project where you brought a drug to help patients. And he looked at me and said, yeah, I can see that, Ian. Plus, I didn't really want to write grants, and that just wasn't my thing. I wanted to do applied research. That philosophy of wanting to develop drugs and help patients has carried me through my entire career. And I've been lucky enough to be involved with over my course of career at Bayer and at Pfizer, where I went after Bayer shut down in Connecticut, and I was in oncology drug development. So at Bayer, I was in research, and then at Pfizer, I was in drug development, so clinical trial. And I've been lucky enough to be involved in 6 programs that have made it to the market. That's really fortunate. A lot of people work their whole career, and they're not involved in one that gets to the market. And I've always been drawn to oncology. Now at Arvinas, we're at neuroscience as well, because of exactly what you said. You tell people you're working in cancer, and immediately can resonate with that, right? Because everyone knows cousin, a grandmother, a nephew, any neighbor who's been touched by cancer. And same with neurodegeneration with Alzheimer's becoming more prevalent. And so it's been gratifying to be able to, connect with people with the research that you do and trying to help, maybe not their spouse or their family member, but the next generation. And really, we're trying to cure cancer. We're trying to cure these neurodegenerative diseases. That's really hard to do. But we're really trying to extend people's lives, make these diseases more, but improve their quality of life, not just their length of life. Kind of turn it into like diabetes or something, where you can take a pill and control that disease for a really long time. And that would be a success, right? For people to make that next graduation or the wedding or whatever, that next family milestone. That's constantly what I strive for and I think about. Now that we're a public company, there's a lot of focus on investments and the stock price and whatever. I always say to the folks here, focus on the patients, the rest will take care of itself. And that's been the thread throughout my whole career.
Yeah, that's very sound advice. In the corporate world, we hear when you focus on your customers, you can't really miss. You can't really go too far because you're very customer obsessed is a phrase I hear a lot in the technology space. So with that as a background, thank you for going through that career history there. That's really fascinating to hear how people get to where they are. What are some of the guiding principles that maybe govern your day-to-day as the chief scientific officer?
It's high quality science is the first thing, right? Making sure that everyone's doing experiments the way they need to be done so that we can really interpret our data, develop the best compounds we possibly can. So it's all about designing the right experiments to answer the next critical question. And of course, to do that, you need to hire great people and surround yourself with great people. I think that's common to almost any organization that's really great is oftentimes the people that surround the leaders, whether it be a CEO or CSO, are actually smarter than you are, but need leadership and to really excel. And so I always say that my job is to make the people in the research group, their job easier, remove barriers, allow them to be creative, innovative, do the best possible work that they can do. Because they're actually doing the work. I don't do any real work. I review data, I give suggestions, but I'm not in the lab anymore, which I think everyone is thankful for, doing the experiments. You gotta enable the people who are actually doing the work to do it the best that they possibly can. So remove barriers, give them advice, give them leadership, give priorities, make decisions. That's what I've learned throughout my career. A lot of times people just want somebody to make a decision and have it be clear, understandable, and they're like, okay, they may not agree with it, but at least they know, okay, this is what the priority is, this is the decision, this is the direction we're going. And so that's how I approach it. Hire great people, point them in the right direction, remove the barriers, and then get out of it.
Yeah, that's very Sound leadership advice. And with hiring great people and where Arvinas is today, and my mind's still blown at the potential of all the technology that you're working on over there. So much has happened over the last decade with technology advancing and just the tools to be able to research, do high quality research like you're saying. Where do you see, if we look towards the future, where do you see the next 5 to 10 years going in the space? Maybe not just for Arvinas itself, but also just in the field overall. Where do you see things evolving?
Yeah, targeted protein degradation, I feel like in general, we've just scratched the surface. I mentioned Craig Crews, the Yale University professor who started the company, published the first PROTAC. I said to him probably a year or so ago, I said, Craig, with PROTACs, you've not just opened up a new therapeutic modality, new approach to treating these diseases. We've opened up a whole new area of science, right? Understanding the ubiquitin proteasome system, how to hijack effectively. And it's really spawned this whole other area of what's called induced proximity. So with a PROTAC, we're inducing the proximity of our target protein in the E3 ligase to lead to degradation. But you can, and now companies are expanding that beyond protein degradation to other enzymatic functions like phosphorylation or dephosphorylation, acetylation. People have now taken what we've done with targeted protein degradation and bringing it into other areas. And it's just expanding like exponentially. This whole idea of hijacking normal systems that the cell uses to basically modulate disease-causing proteins and therefore be able to treat diseases. So that's where I see it going. Where we are in the next 5 to 10 years is moving beyond degradation, where, as I said, companies have flooded in. So now going to these other processes that you can hijack and different ways of then treating diseases. And ultimately combining those approaches, right? Certainly in oncology, the way cancers are treated now, it's rarely by just a single drug. It's multiple drugs. You always have to do your clinical trials and that kind of thing with single drug, but ultimately you get to combinations because I always say cancer cells are smart. They figure out they're trying to stay alive, and it's accelerated up. They have new mutations, new ways of bypassing the pathway to stay alive. And they're trying to stay one step ahead. It's the same as if you're trying to stay alive, you're going to do everything you can to stay alive. Well, cancer cells doing everything it can to stay alive and trying to kill it. So that's why you need to take multiple avenues. One avenue could be targeted protein degradation, another could be dephosphorylation by induced proximity. So that's where I see it going, just taking many different approaches to ultimately, as I said, make cancer either, it's hard to cure cancer. That's our goal, but to make it more of a chronic disease and have people live 10, 20, 30 years with a cancer and just take their medication and live their life as fully as they possibly can and not have this terrible side effects here with chemo and debilitating and not wanting to get out. That's what high quality of life, extended life. That's the goal.
That's a glorious vision for the future, is to have that as a possibility. And it seems within our lifetimes that with how things are going, for the first time, it feels like human ambition and technology are starting to intersect, and we could see it.
Yeah, listen, artificial intelligence is a hot topic right now. We use it at Arvinas. But the power of what you're seeing with artificial intelligence technology, where you're going beyond, you're now seeing things that a human couldn't see, that you help using computer networks to analyze data, see trends that we wouldn't be able to pick up on our own. That's obviously being applied all over the place, including in our work. So that kind of speaks to what you're saying. I mean, that's really going to, I think, push the evolution of technology even quicker, bring therapies to market even quicker, help patients, again, coming back to that, helping patients even more.
Mm-hmm. Yeah. You mentioned artificial intelligence really quickly. Just a quick side question here. I'm curious, we at Qualio, we provide a quality management system to help people meet certain federal regulations and qualities of standard. What at Arvinas, what does your technology stack look like? You mentioned artificial intelligence. What are some tools or other things that you're using? You don't have to go into any, I guess, too specific if there's any competitors listening to the show. But like, what's your normal tech stack? What is it that you're using? You mentioned AI. Are there machine learning things you have that are helping you advance not just the core technology but that you have there, but supporting technologies?
Yeah, so we have a lot of technologies. I mentioned proteomics before, right? So this is where you take cells and grind them up and run them through mass spectrometers and see all the different proteins that are there, and hopefully only degrading the one that you're going after. We use a lot of bioinformatics, a lot of computing power. So bioinformatics, data analysis, computational chemistry. So we're in the cloud. We have chemists who basically do virtual screening. So you'll take a structure of a protein. You'll know what the structure of a protein is, and try to dock molecules in there that are warheads for ultimate PROTACs. So we do virtual screening. So you don't have to actually do real screening. You can do virtual screening with a whole library of compounds. It's just a way to go really much faster. Now, it's always supplemented with what I'll call traditional biology, traditional medicinal chemistry. In the end, you have to test these compounds in cells. You have to test them in tumors. You have to test them in various different assays. You still have to have a chemist make the molecule ultimately, so that you need good medicinal chemists who can understand, okay, we'll look at the data and say, all right, I made this change to the molecule. It had this effect. So now I'll make this change and make it even better. So there's always that fundamental chemistry and biology that's there, but a lot of computational chemistry, proteomics, and that a lot of it is understanding how to design the PROTACs to bring together that target protein and the E3 ligase. So we'll do structural biology. We'll make crystal structure. How do they actually come? So actually be able to see in 3 dimensions, how are those coming together? Because how they come together is really key. You want it to come to efficiently together, so you get the most efficient ubiquitination and therefore the most efficient degradation. And then if we make a change to the PROTAC, how does that change it? Is it making it more efficient? So structural biology, being able to see that in 3 dimensions is really key. Again, supplemented with all the computational power that I mentioned. So that's our stack. We've got the fundamental aspects of biology and medicinal chemistry that's always there, but all the other inputs with the various technologies I listed to try to make that process more efficient, less empirical, less trial and error, so that we, again, get these drugs to patients and into clinical trials faster and faster.
Yeah. It seems like the timeline from getting into clinicals and then out of clinicals seems to be shortening because of these accelerating technology.
That's true. That's part of it. Yeah, because I didn't mention like genomics, where we're finding the true driver mutations, for example, in oncology, they're causing tumors. So that targeted therapy, that personalized medicine that you've heard about, precision oncology, that really helps because you can take it out, sequence its DNA, find the mutation, or even take somebody's blood and just look at the DNA that's floating around that's been released from the tumor. That's even easier than taking a tumor biopsy. Just draw some blood and do the sequencing. So being able to identify those driver mutations, design drugs to them, pick those patients out, right? Before precision oncology, you'd be testing basically all comers, but maybe your drug only works on a selection of those. Now being able to identify those up front, fewer patients are treated, trials are quicker, they're cheaper, quite frankly, that helps. And then you can get the clinical trials done much quicker. So what used to be 12 years could be down to 3 or 4. That's a huge difference. Absolutely. Yeah. If somebody's listening to this today, you could potentially, in the next 5 years, be treated by something we're talking about today. That's just without question. Yeah, that's just fascinating. This podcast is all about going from lab to launch and talking to the innovators in the life science space. So Ian, this is a fantastic conversation. Thank you so much for coming on the show today and sharing some of the insight behind the scenes of what's happening at Arvinas and just this field of study. I feel excited. I feel there's a bright future for what's going on there. And anybody else that's listening, hopefully you feel the same. You can't really see Ian. I can see him because I'm talking to him here, but you can just see the passion and how you're talking about these things. Before we wrap up the episode, we close with one question, which is if you, Ian, could leave a voicemail to every person in your industry or in your field, some advice or something to help them continue to advance the field forward, and a voicemail that everybody would listen to, what would you say? I hate to be repetitive, but I guess I will be, because it's what I would say. It's the same thing. So it's focus on the patients. The rest will take care of it. Yeah. Sound awesome. Okay. Ian, this is, again, like I said, this has just been a fascinating conversation to have you on the show. Thank you so much. If people wanted to get involved or get in touch with you, I know that at Arvinas you have a pretty robust partnership program, but if anybody wanted to get in touch with you or with Arvinas, where can they go to get? Yeah, go to the website, or I think there's definitely some links there that you can get in touch with, whether it be investor relations or just a general inbox, or they can just email me directly, ian.taylor@arvinas.com. Simple as that. Okay, awesome. Thank you for offering that. Ian, again, thank you so much for joining the show today. We wish you all the best. Looking forward to following what you're doing at Arvinas, you and your team. Fascinating technology with real, everyday applications and potential to save lives or extend life, which is something that I think should get everybody's attention and interest. So thank you again so much for joining, and wish you all the best. Yeah, this was really fun. I really appreciate the opportunity.
Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time.